Hello everyone. In this lecture, we'll be talking about ribosomes and mitochondria. Let us begin with ribosomes. We all know that ribosomes make proteins, and the proteins are those molecules which are essential for many cellular functions, ranging from damage repair to directing chemical processes. When we try to locate ribosomes within a cell, they can generally be found floating within the cytoplasm or attached to an endoplasmic reticulum. Such an endoplasmic reticulum that is associated with ribosomes or that is attached with ribosomes is called the rough endoplasmic reticulum. And essentially, this location of the ribosome indicates what kind of a protein it synthesizes. Where the floating ribosomes produce those proteins that will be utilized within the cell, whereas the endoplasmic reticulum attached ribosomes are responsible for the synthesis of those proteins that may be used inside the cell or exported to the outside of the cell. Coming to the structure of ribosome, generally ribosome forms a complex with RNA and protein. Such a complex is known as the ribonucleoprotein, and this kind of a complex is essential for the purpose of protein synthesis. And a ribosome will be composed of two subunits, which are generally known as the smaller subunit and the larger subunit. The functions of these subunits are also independent but equally associated for the purpose of protein synthesis. Like, for example, the smaller subunit has the function of binding the mRNA and decoding it, whereas the larger subunit is where the amino acids get added to form the protein. Whatever it is, both the floating and endoplasmic reticulum bound ribosomes will have similar structures. There won't be any difference in the structure of the ribosomes no matter what their location within the cytoplasm is like. However, there is a difference in the structure of the ribosomes that are found in prokaryotes and the eukaryotes. In the prokaryotes, we generally find the 70S ribosomes, which are comprising of two subunits known as the 30S subunit and the 50S subunit, where the 30S subunit is the smaller subunit and 50S subunit is the larger subunit. Similarly, in the eukaryotes, we have the 80S ribosomes, which comprise of the smaller subunit, that is the 40S subunit, and the 60S larger subunit. So by now, the question that arises here is, what is S that is mentioned here? Well, the S stands for Swedberg units, which is an indicator of sedimentation coefficient. meaning that it indicates the rate at which these ribosomes or ribosomal subunits can settle to the bottom of a tube when it is centrifuged. Furthermore, each ribosomal subunit will have three binding sites. First one is known as the A site or the amino acid here. Okay, the same thing can be found here also. And this is the amino acyl site. And this is the site that accepts the incoming amino acyl tRNA. Then now this is followed by the P site or the peptidyl site that holds the tRNA with a nascent peptide chain. A nascent peptide chain is that chain which is currently being built. So, at the peptidyl site or the P site, the tRNA is transferred, and here the amino acid will be attached to the growing peptide chain. And then is the E site or the exit site, where the tRNA that has just been removed with the amino acid, or it is known as the deacylated tRNA, exits. So this is the place where the exit of the deacylated tRNA happens. So these are the three sites that are commonly found in all ribosomes, and it plays a crucial role in the functioning of that particular ribosome. Next, talking about the functions of ribosomes, there is only one important function that is carried out by a ribosome, and that is assembly of amino acid to form proteins. And this is a very essential process to carry out cellular functions. The DNA generally produces mRNA by the process known as transcription. As you can see here, there is an mRNA that is formed, and this process is known as transcription. And then this mRNA, which is synthesized in the nucleus, always remember transcription happens in the nucleus only because DNA is found in the nucleus. So, the product of transcription, mRNA, which is synthesized in the nucleus, is transported to the cytoplasm for protein synthesis, because ribosome cannot be found inside the nucleus, and ribosome is an essential cell organelle required for protein synthesis. So the mRNA is sent outside the nucleus for this purpose, and then the ribosomal subunits attach to the mRNA, and the tRNA is recruited to carry the amino acids required for the protein synthesis. And this process is what we call as translation. So this is how ribosome works within a cell. Nextly, we have the mitochondria. Mitochondria in plural, or it is singularly called a mitochondrion, is a double membrane bound cell organelle, which is typically of a size ranging from 0.75 to 3 micrometers. Mitochondria is found in most of the mammalian cells except for mature RBCs. You should also remember that the mature RBCs will not have nucleus. Okay, so mitochondria or nucleus are not found in mature RBCs or the erythrocytes. And these mitochondria are regarded the powerhouse of a cell, mainly because it is the site of ATP synthesis, meaning that it plays a major role in breaking down nutrients to generate energy rich molecules for the utilization of the cell. So, in layman terms, mitochondria is like a digestive system of the cell, and also the biochemical reactions that are involved in the cellular respiration will occur inside the mitochondria. So, talking about mitochondria, the word, it is derived from Greek. Okay, it is a combination of two words called the mitos and the chondrion, which mean thread and granules like respectively. And this was a cell organelle that was first described by Richard Altman, who is a German pathologist, and he described this mitochondria back in 1890 itself. So now coming to the structure of mitochondrion, as I told you already, mitochondrion is double membrane, rod shaped, and it is found in both plant and animal cells. Talking about the diameter of a mitochondrion, it will generally be of a range from zero point 0.5 to 1 micrometer, and it will comprise of three components, namely the outer membrane, the inner membrane, and the matrix. Outer membrane and inner membrane are membranes, whereas matrix is a gel-like material that fills this cell organ. So when we say that the outer membrane and inner membrane are membranes, they are essentially made up of proteins and phospholipid bilayers. However, they are separated from each other, leaving some space in between. And this space that is found between the outer membrane and inner membrane of the mitochondria is called the intermembrane space. And with specific reference to the outer membrane, it will have a large number of special proteins known as the porins, which will make the outer membrane permeable to ions, nutrient molecules, energy molecules, etc. And the most commonly used energy molecules in this regard are the adenosine diphosphate and the adenosine triphosphate. Now, moving on to the inner membrane of the mitochondria, you should know that it is a complex structure. It is not as simple as the outer membrane. Why? Because it is found folding into multiple layers on the inside of the mitochondria to give rise to a structure called cristae. This kind of a folding happens mainly to increase the surface area that is found occurring inside the cell organelle. So, when there is an increased surface area, there is larger space for the biochemical reactions to occur. So, what happens here is the inner membrane proteins, when coupled with the cristae, they help in the production of ATP. And you should remember that the inner membrane is strictly permeable only to the oxygen and ATP molecules, whereas the outer membrane was freely permeable to ions, to the energy molecules, etc. Okay, that is not the case here. Okay, so most of the unwanted molecules are filtered out by the inner membrane, and only those molecules required for ATP production are let inside. Next, we have the mitochondrial matrix, which is a viscous fluid that contains a mixture of enzymes and proteins. Furthermore, it will also comprise of ribosomes, inorganic ions, mitochondrial DNA, nucleotide cofactors, and organic molecules. Essentially, these enzymes that are found in the matrix are present to aid in the synthesis of the ATP molecules. So looking into this diagram, you can see that we have a mitochondria here, and there will generally be an outer membrane and an inner membrane. The inner membrane can be found folded to give rise to what is known as cristae, and then we have the matrix space that is enclosed by the inner membrane, and there is another space that is found between the inner membrane and the outer membrane. Also, you should remember that mitochondria will have a DNA of its own, and this is known as the mitochondrial DNA or the extranuclear DNA. So now looking into the intermembrane space and looking at the structure of the outer membrane and the inner membrane, you can see the presence of porins only in the outer membrane. And these porins will allow molecules to freely enter into the intermembrane space. And here, the inner membrane will filter the molecules and pick up only those molecules that are required for ATP synthesis. So basically, this is how the energy production happens within a mitochondria. Talking about the functions of mitochondria, first and the foremost function of mitochondria is energy production through the process known as oxidative phosphorylation. And apart from that, mitochondria will also be responsible for regulation of the cellular metabolic activity, which is an obvious thing because there is requirement of energy for a lot of metabolic processes to occur within a cell. So if the mitochondria does not produce energy, then it is quite obvious that the cellular metabolic activity is greatly affected. So directly or indirectly, mitochondria will play an important role in the regulation of cellular metabolic activity. Nextly, mitochondria helps in promoting the growth of new cells and cell multiplication. This is essentially due to the production of energy, and when there is more energy, the cell will have capacity to replicate or form new cells, etc. Furthermore, mitochondria helps in detoxifying ammonia in the liver cells. Ammonia is an essential toxin that is commonly found in animal cells, or that is commonly formed in animals. So, and you know that liver is the organ where most of the detoxification processes occur. And liver cells are highly populated with mitochondria for this purpose of detoxification. And mitochondria majorly helps in detoxifying ammonia. Also, mitochondria plays an important role in apoptosis or programmed cell death. As I told you, mitochondria can stop producing energy if it wants to kill the cell. It's that simple. And mitochondria can also be responsible for building certain parts of the blood and various hormones such as the testosterone and estrogen. These are mainly the steroid hormones, testosterone and estrogen, and cholesterol metabolism is carried out in the liver cells by mitochondria. So, it is only when cholesterol is broken down can these molecules be formed. So, mitochondria plays an indirect role in synthesis of these steroid hormones. Additionally, mitochondria helps in maintaining calcium ion concentration, and it is also involved in various cellular activities like cellular differentiation, cell signaling, cell senescence, regulation of the cell cycle, and also the cell growth.
Yeah, so that is it for this session. Thank you all. If you have any doubts, you can always write an email to me.